Happy Halloween.
The zombie apocalypse.
Zombies.
Zombies. When zombies start to take over the world, that's going to be a real problem.
They're not very healthy.
We train every day for the zombie apocalypse.
Takes you back to the bare bones of medicine. You won't have as much tech, right?
My hope is that we'll have a greater understanding of how we can get rid of them.
We will have specific weapons that will target from our belts.
I own shovels, which I've been told are great.
There's a zombie. I didn't know it was there. Bam, he's gone.
I do have food and supplies and a generator at the house, so everyone's welcome. But every day we're going to work to prevent it, so hopefully none of that will be needed.
Well, good morning. Happy Halloween and welcome to our spooky science in the city. We hope you enjoyed that lighthearted look of zombies from several of my colleagues here at UT Southwestern. My name is Dr. Stuart Ravnick, and I'm an Associate Dean at the UT Southwestern Graduate School of Biomedical Sciences and Assistant Professor in our Cell Biology Department. From our entire planning team of this event, we are so happy that you are joining us this morning. We appreciate it. This is the third year of Science in the City, and it has been fun for me to be involved in this program since the beginning. And even though we can't gather today in person for lab tours, we can take you on this virtual tour. So created by the Dallas Morning News, Science in the City is a family-friendly special event that connects the Dallas-Fort Worth community with local researchers working at the frontiers of their fields. The program is a unique collaboration between UT Southwestern, Dallas Morning News, Southern Methodist University, talkSTEM, the Center for Brain Health, UT Dallas, and the Perot Museum of Nature and Science. We are so proud to partner with the news on this behind-the-scenes tour right here at UT Southwestern. First, a little bit about us. Did you know that UT Southwestern is one of the premier academic medical centers in the country and the only one in North Texas? This means that we have a three-tier mission of research, patient care, and medical education. Also, many people don't know just exactly how large we are in terms of a powerhouse of education. We have the UT Southwestern Medical School that trains nearly 950 physicians. We have the UT Southwestern Graduate School of Biomedical Sciences, which trains nearly 500 scientists and then postdoctoral fellows. And we have our School of Health Professions, which trains about 400 students in those professions. Among our number of honors, UT Southwestern is proud to have 6 faculty members who have been awarded the Nobel Prizes. Our faculty of more than 2,700 is responsible for groundbreaking medical advances and is committed to translating basic science-driven research to new clinical treatments. So back to today. We're going to take you through a journey through some of this research in science. We're going to learn about our electric brains, to practicing some spooky yoga, to exploring how we use cellular glow sticks to highlight things in our bodies. And we hope you enjoy our spooky science. We do have a few technical directions, and I want to remind you, please keep your microphones muted during the presentations. This helps everyone to hear them. We will have question and answer sessions at the end of each of our lab tours. During these sessions, we're going to ask you to use the raise your hand function in Zoom. And to do that, down at the bottom or somewhere on your Zoom screen, you'll see the participants on your menu. Click the 3 dots on the bottom of the participant screen and click raise your hand. We look forward to your talking. In fact, if you have a question, we'll call on you to turn on your camera and unmute your microphone. Obviously, if you have any technical problems, put them in the Zoom chat box and we'll try to help you. And with that, let's pass it over to Anna Kuchman, our partner and science reporter at the Dallas Morning News. Thank you so much.
Hi, I'm Anna Kuchman, science reporter at the Dallas Morning News. Happy Halloween and welcome to our 3rd annual Science in the City. The Dallas Morning News started this program in 2018 to connect the community with local researchers working at the frontiers of their fields. As a science reporter, it's my privilege to go behind the scenes into research labs, spend time with scientists, and learn about their work. Science in the City lets us bring that firsthand experience to you. Normally, we'd all be meeting face-to-face, and I'm sorry we can't do that this year. As you can see, I'm at home with all kinds of strange creatures. But we felt that this was an especially important time to highlight and celebrate science in our community. We're grateful to all our partners for coming together and making it possible for you to experience these events even under these unusual circumstances. This year, Science in the City is a series of 5 events. Our next one is Brain Health in the 21st Century, the Next Frontier on November 5th. On November 7th, we have an exploration by the nonprofit talkSTEM of math and science in novels, architecture, nature, and even junk food. And on November 14th, you'll have the chance to meet SMU physicists who study black holes, dark energy, and other exotic phenomena in the universe. You can register at scienceinthecity.dallasnews.com. All the events are free of charge. We are so grateful to UT Southwestern for creating Spooky Science and hosting all of us today. If you enjoy this series, please consider supporting the Dallas Morning News with a subscription at join.dallasnews.com. You'll be the first to hear about events like these. Thank you.
Thank you so much, Anna. It's great to have that welcome. And now I'd like to pass it on to Dr. Mark Goldberg, who's going to take us through a tour of our electric brains.
Thank you, Stu. Thank you, Anna. Welcome to Science in the City. I'm Dr. Mark Goldberg. I'm a neurologist and neuroscientist at UT Southwestern, and each year I have the privilege of guiding our visitors through some amazing labs at the Peter O'Donnell Jr. Brain Institute. Our topic today is our electric brains, and pretty soon we're going to be visiting a facility with an amazing new machine that allows us to detect the smallest electrical impulses from living human brains, just like each of ours. The signals that make up the circuits that help our brains when we move or when we dance, when we think, when we learn, when we feel emotions. And to help us understand that, I'd like to have you join me in an experiment. We're going to do this together. Are you ready?
Hi, my name's Katie. I was the zombie in the video. I have actually worked for many years as a zombie, professionally. Now, however, I'm a graduate student working on my PhD in neuroscience. I study how the brain recovers after injuries like stroke. One of the things that I've found in my research that constantly amazes me is the ability of the brain to rewire and make new connections after there's been injury.
Hi, I'm Lauren. I'm a technician in the neurology department. I study blood vessels in the brain, and outside of the lab, I'm a huge horror movie fan. And I started thinking, why are our brains wired to respond to horror movies in that way? And why do we find them scary? Why is it fun? So I made this video so we can start thinking about that.
So that's our experiment. What did you observe about how you felt as you watched the film? Did you notice there at the very end, suddenly there's a big reveal and there's a jump scare at the end? And our bodies are filled with adrenaline. And if you were being chased by a zombie, you'd be ready. That's the fight or flight reflex, and our brains generate that. How about as we're walking through the lab, we pass many things, just normal laboratory items that aren't scary at all, test tubes and shakers, things like that. And yet, we've noticed that something about that scene makes us know there's a frightening thing just around the corner. How is that? Our brains have learned that in a horror movie, these sound effects, this frightening music, or this lighting changes or fog. All of this mean the same thing. There's a threat ahead. And our brains have learned to put those things together to trigger an emotion, to anticipate that something might be happening. And if you were in the zombie apocalypse, that would be useful information to have. Well, to understand how our brains put together memories and process emotions, as an example of the kind of work we do, we're going to visit the Magnetoencephalography, the MEG laboratory, and we're going to look at some human brains. And to start us with this, we're going to ask Dr. Amy Proskovic, who is a neuroscientist who works in this area, to help explain what we just experienced. Thank you, Amy.
All right. So I'm sure you guys have been scared or spooked at some point in your lives, am I right? Yeah. All right, so you probably noticed that when you get spooked by something, say a zombie coming after you, a couple things happen in your body. So your heart is probably gonna start pounding, and you may get goosebumps and feel like your hair is raised on end. And you also may get this surge of energy that kind of helps spring you into action if you need to either fight that zombie off, or if you wanna run and get the heck away from it. So that's your body's fear response happening, and there's actually a network of different brain regions that help get that fear response going. So the main area that really underlies that sense of fear in the brain is called the amygdala. And so it's a structure that's in the middle of the brain. And so if you look over here on the screen, this is looking at the side view of the left half of your brain. And so if I rotate it, then the structure that you see that's colored red is your amygdala. So when you get spooked, your amygdala gets really excited and is really active. There's also some other areas that underlie fear. One thing that's important is if you are in a zombie apocalypse, it's probably good for you to remember where you encounter a zombie or other things that might clue you into, hey, a zombie might be present. And so an area that helps you kind of learn and remember fearful events is this guy here in blue. So that structure is your hippocampus, and that helps you learn and remember fear. And then, of course, hopefully you'll be able to get away from the zombie if you need to and calm down. So you no longer need to be afraid at that point. And the area of the brain that helps regulate your amygdala and that fear response is called the medial prefrontal cortex. And so that is shown over here in this teal color. So that area helps regulate your fear response and say, hey, there's no longer a threat present, I'm okay. All right, so now that we saw on the computer the brain areas that are involved in the fear network, we actually have some real brains over here that you guys get to interact with. All right, so over here you can see this is kind of the real version of what we were looking at on the computer screen. So the amygdala would be in this general area, and then the campus that helps you remember is kind of behind the brainstem there. And then the medial prefrontal cortex that helps you regulate your fear is this area up here. So if you guys want to touch these, you can hold them, you can see how heavy they are, what they feel like.
Can you feel the mouth on Lucy? Put your hands under it and you can hold it. No? You can hold it. Hold this one? No?
All right, so one way that we're actually able to non-invasively look at how the brain functions is by using a type of neuroimaging called magnetoencephalography, or MEG, or MEG for short. And so I actually have our MEG machine behind me here. And so this is a state-of-the-art neuroimaging device. It is the newest model available, so we're very fortunate to have it here at UT Southwestern. And what MEG does is it measures your brain activity millisecond by millisecond. So your brain is always active and it works and processes information at an extremely rapid pace. So MEG really stands out compared to other types of brain imaging because we're able to capture that brain activity on such a fine temporal scale. Another awesome thing about MEG that sets it apart from other types of neuroimaging is that it has good spatial precision. So we're able to localize where in the brain is active within a few millimeters. So that combination of being able to record activity really quickly and with good spatial precision is what makes it an ideal tool for studying human neuroscience and behavior. All right, so now that we have Lucy in there, we are able to look at her brain activity. And so over here on the screen, you see all of these lines, and each line is from one sensor in the helmet in the MEG. So each line shows the sensor data, and we have 306 sensors overall recording her brain activity right now. And MEG records her brain activity millisecond by millisecond. So it has a really excellent temporal precision. And another awesome thing about MEG is that it has good spatial precision. So after we bring her out and we do a little bit of processing on the data, we'll be able to localize where in her brain is active within a few millimeters. And really state-of-the-art method for studying human neuroscience and behavior.
Hello, and that's our presentation about the brain. And I would like to join here Amy Proskovic, and we'll be here available if you'll unmute to answer any of your questions. Okay, and as we're getting started, let me ask the audience and all of you together. Amy, it's breakfast time almost. Which would you rather eat if you had to eat? Something from this presentation, jellyfish? Would you want some worms to eat? Or maybe some brains would be delicious. What do you guys think? And I'd like to have your questions too. Let's have your questions about what we just heard. What would you like to ask us about scary movies? What would you like to ask us about looking inside the brain? And what is this outfit? Dr. Amy, what are you?
So I am a rainbow tiger for Halloween this year.
Okay, I've got one vote for jellyfish. Would that be strawberry or grape jellyfish? Okay, thank you for that vote. Okay, anybody have any science questions for us? And you know, to ask a question, just put that in the chat. And
Goldberg, we do have a question from Tammy.
Let's hear it. I am so ready.
Tammy, if you want to go ahead and unmute yourself, we would love to hear your question.
Okay, so is the brain really squishy, or and the in the video, was that an actual real brain?
Okay, Dr. Amy.
Yeah, so that really was a true human brain. So we're very lucky that we have people who volunteer and donate their brains to science. So that helps us learn a lot of things about the brain that we wouldn't be able to do if we didn't have those awesome people who help us out in that way. And so we had real brains, and we had some of our friends from the community come in and get to experience what those brains feel like and how heavy they are. And they are a little bit squishy, you're right. But yeah, it was a fun experiment.
And we have another question. Was there another question for us? Well, this has gone by very fast.
Sorry, I'm going to interrupt you for 1 second, Dr. Goldberg. We do have one final question from Otakar. Otakar, if you want to go ahead and unmute yourself.
Let's have it.
All right. Giving Otakar just a second to unmute.
Sure, you go right ahead. And oh, go ahead, Otakar.
How does your brain heal things?
How do brains do what?
Hear things.
How do brains hear things? Okay, Dr. Amy, this is for you.
How do they hear things?
Yeah.
So there are a couple different areas in the brain that help you hear. And the main one is it's kind of right where your ear is. So it's on the side of your brain and it's called your auditory cortex. And so you have one on the right side of your brain and then one on the left side of your brain, kind of right behind where your ears sit. And those really help you hear and be able to understand language and other things like that.
What a great question that is. And we have just one more question, time for one more question. So it looks like Josephine has got her hand on the screen. Hand up. Josephine, do you have a question for us? Christian would like to know what happens to a brain during a headache. Let me say that so would we like to know that. That's an excellent question. And that is a question which has challenged neurologists and neuroscientists for years and years. It's really hard to know what happens to the brain during a headache. Does the brain feel the pain? Of course it does, but is the pain coming from somewhere outside the brain, like the layer of the fine membrane around the brain or in the blood vessels? And believe it or not, we just don't know exactly. We know where the pain receptors are, but we don't know what's going on when you have a headache, depending on what kind of headache you have. So it's a real challenge, and it's really hard to imagine when you're having a bad headache, something's going on in your brain. Well, I just want to go ahead and think, I just want to say, I am a brain person, so the answer is certainly to eat the brains. And I hope I'll share some with you if you want to have a little bit, but certainly I'm ready to have a great big, delicious brains. And for those of you who are participating here online, the recipe for these brains, peach, these brains is in the online materials for Science in the City. And with that, we're going to turn to the next activity. It's spine tingling yoga. We're going to the Spine Center, UT Southwestern Spine Center in Frisco, and away you go. Thanks, everybody. It is Jello. Yes, it is. Okay, not real brains. We have real brains too, but delicious.
Thank you, Dr. Goldberg. We will have time for the additional questions in the general Q&A, so I am going to pass it to the spine group.
Awesome.
Hello, everyone. Welcome. The spine is so important. It supports the entire body. It supports the entire body, and it's important for us to take care of it.
Hey, Corey, can you unmute, please? We lost the sound.
Start to rock side to side. Massage the low back. And then you can pull your right knee into the chest. Extend your left leg long to the ground. We'll take a supine twist, bringing the right knee over to the left. Tee out your arms long. Bring both shoulders to the ground and gaze over your right shoulder. This will help lengthen your spine. You can do this pose at home. And when you do this, start to deepen the breath, taking inhales to lengthen your spine and exhales to twist just a little bit deeper. And stay in this pose for about 3 to 5 breaths. And once you've done that, you can come back to the center, pull both knees back into your chest, and make sure to get both sides. So we'll switch to the left. The left knee will stay hugged in. Extend the right leg long. Bring the left knee now over to the right. Keep your arms long. Your gaze is going to go to your left shoulder. And you can hold this pose. It'll help lengthen the spine, lengthen out the muscles in this back. And hold again for 3 to 5 breaths. And once you've done this, come back into the center, pull both knees into your chest. To warm up the spine further, you can grab behind the thighs and take a few rock and rolls up and down the length of your mat. You can be on the carpet or on the grass. Any place is good for yoga. This will help warm up the spine. The next pose I'm going to show you is called cow and cat. This will also help mobilize the spine, warming up the joints. To take this pose, bring your hands flat onto the ground. Your pointer finger is going to be forward. Your legs are hip width distance. Your arms are shoulder width apart. Shoulders are just above the wrist. Hips are just above the knees. To take the cow pose, you're going to drop the belly and gaze up to the sky and take an inhale here. As you exhale, round your spine and go into cat. You can take these poses through your exhales and your inhales, help mobility in the spine. And I'll just show one more pose real quick. It's going to be a forward bend. You'll bend one knee, come into the right leg and reach over. And we're running out of time, but these are just 3 poses that you can do at home to help strengthen your spine and improve mobility. I'll now pass it over to the spine team at UT Southwestern Frisco.
Thank you so much. Those are great exercises for all of us to learn how to do. I'd like to introduce myself. I'm Dr. Nasser Ayed. I'm one of the spine physicians at UT Southwestern Frisco. And we have a group today, and our goal is to do a couple of things. First of all, we'd like to introduce ourselves, and then we'd like to talk a little bit about some basic spine health, some practices, and some do's and don'ts. So I'm going to pass it off to Dr. Daltrey Dot to get us started.
Hi, thank you for joining us this morning. I'm Daltrey Dot. I work here at UT Southwestern Frisco in the Spine Center. We're located at the Tollway and El Dorado, and we offer multiple medical specialties as well as inpatient, outpatient, and radiology care to bring the UT Southwestern excellence and care that you know from downtown up to the community in Frisco.
Good morning. My name is Catherine Hose. I'm one of the spine surgeons here, and we wanted to introduce our spine center specifically to you guys. We're multidisciplinary care here, both with surgeons in neurosurgery and orthopedics, as well as non-operative providers in anesthesiology and pain medicine. We are really proud of our excellence in care, and we are happy to bring everything under one umbrella for you here in Frisco.
Hi, good morning. My name is Alphonse Jose. I am one of the physical therapists here at UT Southwestern Frisco. Our outpatient center offers a variety of therapies, including physical, occupational, and speech therapy. We offer services to injured athletes and weekend warriors, or any cases that you may need physical therapy, occupational, or speech therapy for. We work really closely with the spine center and communicate frequently with the referring physicians. We offer one-on-one service to all our patients. Today, we will be discussing some tips where you can keep your spine healthy and strong.
Okay, so the first thing we're going to talk about is the importance of a very basic activity, something that we do every day, but we need to set some special time for it aside as well, outside of our daily activities, and that's just walking. Walking is a super important thing for all of us to do at least 30 minutes a day outside of just walking to the kitchen or walking to the bathroom. Just go on a walk. We try to walk it. We recommend at least 30 minutes a day, 4 to 5 times a week. Walking does a few things for us. Number one, it helps keep all the muscles that support our spine strong and flexible. So our abdominal muscles, our back muscles, and it helps keeps our legs strong because that's where our spine sits on top of our legs. The other thing it does, it really helps with our circulation and keeps good blood flow moving through the body. And lastly, it just makes us happy. So we highly recommend walking, again, at least 4 to 5 times a week, and try to get 30 minutes in. Of course, always do it with comfort. So keep walking on your list. The next thing we're going to do is we're going to talk about some do's and don'ts when it comes to how to set up yourself when you're at home. And I'm going to pass this on to Alphonse.
With the pandemic going on, we find ourselves at home doing school or work. And this is how we usually sit when we are concentrated so much onto our work. We find ourselves like slouched, leaned forward. So there are some tips that you can do with the sitting posture that you can prevent back pain to develop. So find yourself a good chair that has a good back support and sit all the way to the back of the chair. If you have a small pillow like this, or you can use a rolled up towel in the small of your back, that will hold your spine in a better position where you cannot slouch. You also want to adjust the height of the chair in a way where your feet are supported on the floor. If the chair is too high, you want to find some stacked up books to help you keep your feet supported. You also want to make sure to move the computer close to you, where your arms are close to your body, where you can type and look at the computer parallel, so you're not bending your neck forward. The next thing that we are going to show you is to how to bend forward to pick up things. We find ourselves a lot bending forward from our back and lifting. If you lift this way, it adds a lot of stress on our back, and over time, this will develop back pain. So to help to keep your spine healthy and strong, we recommend bending from your hips and knees, using the big muscles in your thighs to help you lift. Breathe out as you come up and keep your spine healthy and strong. This is something that we see in our day-to-day life. People are on their phones, bending forward and texting and typing away. So what we recommend is bringing the phone or iPad up closer to your face, where you keep your neck straight and you're looking straight onto the phone. This will help to prevent neck pain to develop. If you practice all three things, we can keep our spine healthy and strong. We also like to show you some spine stabilization exercises where you can do these exercises at home, but we recommend if there is any pain, please do not do these exercises. The first thing that we like to show you is called a bird dog, where you get on your hands and knees and keep your spine straight and kick with your arm and leg opposite, and make sure you keep breathing and switch sides. We recommend doing 10 in a row, take a break, and do another set of 10. If there is any pain, please do not do these exercises. The next exercise we like to show you is an abdominal crunch. To do this exercise, you're going to be laying flat on your back with your knees bent, arms crossed across your chest. Slowly come up as you breathe out and go back down, nice and slow. You can do 10 of these in a row and take a break. Another set of 10 will keep your spine healthy and strong. The third exercise we like to show you is called a plank, where you lift your body up on your elbow, keeping your hips and knees straight. You can hold this position up to 10 seconds while taking deep breaths. If this is too hard for you to do, you can keep a bend at your hips and knees. This is the easy one to do, and you can hold it up to 5 to 10 seconds while making sure you're breathing deeply.
Thank you so much, everyone, for joining us today. We're here at the UT Southwestern Spine Center in Frisco, looking forward to serving all your spine care needs. We'll also be available at the end of today's program to answer any questions for you.
Thank you.
Thank you to our spine team. That was fantastic. And we are going to do a quick Halloween parade because we do want to see everybody's costumes. But before we do that, I do have one question that I want to ask everybody. So I am going to launch a poll that says, what year will have the next Halloween with the full moon?
You're awesome. You're awesome.
All right. So many of you say 2037, but actually it is tonight. So tonight will actually be a full moon. We won't see it for another 18 to 19 years. And it will also be a blue moon. So go ahead tonight and watch. It is very cool and exciting. And one final thing before we move to our next lab, we really want to see what everyone's costumes are. So if you could tell us, are you dressed in a costume? Turn your cameras on. Let's see all the wonderful costumes. And please, in the chat, put what type of costume you're wearing. Personally, I am wearing a spooky science t-shirt that says, I'm only spooky periodically. Thank you to whoever left.
I've got to see how to do it. I can.
All right. That's awesome that so many of you are wearing costumes. I am now going to pass to Dr. Lisa Dennison, who is going to talk to us about molecular glow sticks. Lisa, take us away.
Good morning, everyone. Thanks for showing us your costumes. That was really fun. I'm Dr. Lisa Dennison, and I'm going to lead you on a journey into some of our laboratories here at UT Southwestern, where you'll get to experience science that glows in the dark. The molecular glow stick you'll learn about in just a moment has been so revolutionary for science and medicine that the scientists who discovered and developed it were awarded the Nobel Prize.
Here's how you do it right here.
On our first stop, we'll look into the microscope at some glowing worms with two UT Southwestern graduate students who are currently earning their PhDs, Priyanka Bhatia and Lexis Taji. You'll get to see what they're seeing live through two different powerful microscopes. So Priyanka, please take it away.
All right, thank you. Thanks, Lisa. Hi, everyone. My name is Priyanka, and I'm a graduate student in the lab of Dr. Jeff Woodruff. I study how eggs protect themselves from aging too quickly. So I use these tiny round worms in my research, which I'll be showing you in a second. Now, for scientists, it's really hard for us to see and find stuff inside a cell. Now, if you were lost in a crowd and your parents wanted to make sure they could find you easily, they might put you in a bright orange vest. Or if you're running at night, you wear a headlamp so that you don't get hit by cars. Or you can even hold up glow sticks in the dark, and that helps you see in the dark. Now, for us, we want to be able to see where stuff is inside a cell. So we put brightly colored molecular glow sticks on the stuff inside cells. We call these fluorescent proteins, and they come in all different colors, red and green, yellow and blue. And these proteins come from glow-in-the-dark jellyfish that live in the really cold, dark waters of the ocean. Now, if we attach these glowing fluorescent proteins to almost anything in the cell that we want to study, we can then put that under the microscope, shine a light on it, and it'll glow in the dark. This lets us see where stuff in the cell goes and the different rooms and factories in the cell that make it work. So I'm about to show you a live view inside a microscope in my lab, and I'm going to show you what a green version of this protein looks like, or GFP, green fluorescent protein. Now I'm going to share my screen from the microscope that I use. And as you can see here, I have some glowing green worms. So I'm going to explain what all of this is. Now, we're looking at roundworms that normally live in the soil. These worms are about 1 millimeter long, so they're very hard to see with your naked eye. And it turns out that they're a really useful model to use in the lab to help us learn about disease and development. Right now, these worms are asleep. They're kind of floating around in liquid, but
what you're seeing here is this is the tail end of a worm, and over here on this side is its head. And these worms have the green fluorescent protein, or the GFP, attached to their DNA. All of the DNA is showing up as these green dots. So we can tell that these in the middle here are eggs. All of the eggs have been fertilized and all the cells are dividing very rapidly. And so every time a cell divides, it creates more and more green dots because it's creating more and more DNA. This helps us see what's happening inside the worm, and it lets us see where the DNA is going. And it's really important to our lab. So now I'm going to hand it over to my colleague Lexis, who's going to show you some similar worms that are awake.
Thanks, Priyanka. So my name is Lexis, and I'm a graduate student in the lab of Dr. Peter Douglas, where I can study how to make worms live longer. The lessons we learn from worms can help us understand how we can live longer too. So right now, I'm going to show on my microscope some worms that are awake. And so, if everyone can see, I have worms that are crawling all over the plate right now. And what you are seeing is this pattern in the background. That's the worms actually making their footprints in their food. And so worms actually eat bacteria. And so Priyanka showed you GFP inside of the worms, and I'm going to show you RFP, which stands for a red fluorescent protein. And so with these worms right now, I can turn on my fluorescent light and I can show you my awake worms that are going around in red. And right now you can see that the red is outlining their bodies, and this outline is actually showing the muscle of the worms. And so you can label everything in the worms with different types of fluorescent proteins. And so right now we're looking at the muscles, and if I move it over, we can get to see the brain of a worm. So you can see this tiny little dot, and that is actually the brain of a worm. And if we change it back, you can see that the worm is very tiny right here. And that is the brain. And so people in my lab use this technique to study a bunch of different things. Specifically, they look at how worms can get concussions and how that can translate into us being able to prevent concussions in humans. And so now I'd like to hand it back over to Lisa to continue our journey into the next lab.
Thanks so much, Lexis and Priyanka. That was really cool seeing those glowing worms in action. A quick note for our viewers before we move on. If you want to spend more time with them in the lab, check out Lexis and Priyanka's science podcast called Incubation Time that you can find on any podcast streaming site. Okay, let's head into our next laboratory. Dr. Maralise Konachi-Sorrell is an assistant professor of cell biology who studies how cancer cells grow, divide, and move around. She's going to tell us how GFP can help us better understand cancer and develop new cancer-fighting treatments. Dr. Konachi-Sorrell? You're still on mute. Can you?
Okay, now I can. Thank you, Lisa, and good morning, everyone. So my lab at UT Southwestern uses green fluorescent proteins, or GFP, which are these molecular glow sticks that you learned about from Priyanka, to study how cancer cells grow. So fluorescent proteins are not only very cool, but they're also incredibly useful. They help us see cancer cells, but not only that, but they help us measure multiple aspects of their behavior, including how fast they grow and how fast they move. So we all know that cancer cells multiply very fast, but for them to do that, they need to make massive amounts of materials or building blocks that make new cells. So cancer cells develop very large and efficient factories that make these materials. These factors are so important that in some cancers, the doctors can tell how advanced the disease is just by measuring the size of these factories. So one of the things that we do in our lab is to label these factories in cancer cells using our glowing marker or GFP, and we look for drugs that can reduce their size and stop cancer cells from growing. So Dr. Mari Carmen Lafita Navarro, who is a postdoctoral researcher in my lab, she's in the lab right now, and she's going to show you what cancer cells look like under the microscope. And she's also going to show you these factors labeled with our green fluorescent protein inside live cancer cells. And this is one of her own experiments. Mari Carmen?
Hello, everyone. So now I'm going to show you some pictures that I took in this microscope that I have behind me of an experiment that I did with brain cancer cells. So these are multiple, wait. Is it not present? Okay. So these are multiple brain cancer cells where I use the GFP label protein to visualize those factories that Dr. Kunachi Sorel was talking about that are so important for the cells to produce the building blocks that they need to grow and to divide. So each blue circle is the nucleus of a cell, and inside that has the DNA, and inside each nucleus we can see in green these factories. So what I did, I treated these brain cancer cells with a chemotherapy drug, and I observed by looking at them under the microscope how these factories behave. So if we zoom in to one specific cell, we can see that in the cells that I didn't treat with anything, these factories in green, they are very large and they are very irregular. And this is an indication that these factories are working very hard, which means they are producing a lot of building blocks, which means the cells are going to be able to grow and they are going to be able to divide very rapidly. And this is a bad thing because these are cancer cells. So when I treat this as with chemotherapy drug, what I see in the microscope now is that these factories, they change their shape and now they are rounded and they are smaller. And this is an indication that these factories are not working as hard anymore, which means they are not producing enough building blocks, which means the cells are not going to be able to grow and they are not going to be able to divide. So one of the things that we can do with these GFP labeled proteins is we can do screenings of drugs. We can see which drugs are inactivating these factories by looking at the microscope, and that can help us narrow down which drugs we should continue working on to see if we can maybe someday use them for treating cancer. So that's our research, and thank you very much for joining us. And now I will hand this back to Lisa.
Thank you. That was great seeing the cancer cells as well. Thank you to all 4 of our scientists for taking us behind the scenes in the lab today for an up-close look at how GFP, the molecular glow stick, helps us light the way to new discoveries in science and medicine. So now we have time for some questions from the audience. Just like before, please use the raise hand button in Zoom so that we can call on you to ask your question. It's hard for us to see because there's so many little video icons. It's hard for us to see if you just wave your hand on the screen. So if you'll just use that raise hand button that you'll find if you open up the participant list, then that'll help you pop up to the top. So I see Phoenix, you're raising your hand. Can you unmute yourself?
So does it permanently damage the worm when you shake it to give it a concussion?
So I can answer that. And it does not permanently damage the worm from what we see right now. I don't really work on the concussion aspect of my lab, but when you shake them really fast, all it does is paralyze them. And so they get flat. They look just like a stick. And then within a couple days, they start moving in their squiggly patterns again.
Wow. Is there like a treatment that you use to, like, if the same treatment that you would use for a human, do y'all try that on the worms?
Yeah, we're trying to. That's what we're trying to figure out in my lab is if we can cure it in the worms or help it in the worms and move it up to different types of models. Maybe we can eventually do that for humans.
I see David has his hand raised. David, can you unmute yourself and ask your question? There's some other hands up as well, but David, if you want to go first, or if you don't have a
Dr.
question.
Dennis, David actually just said never mind. So
Oh,
if we
okay,
want to
gotcha. All right, let's do Eddie. Eddie, are you there?
Yes.
Okay, what's your question, Eddie?
Well, I'm his son. So, okay, so
The camera's the wrong way.
a concussion is when the brain hits the side of the skull, right? So then does that mean that a worm has a skull?
The worms do not have a skull. They don't have bones.
Then what makes it a concussion for a worm? What scientifically makes it a concussion?
Well, that's a really good question. We look at that by how the neurons are actually being activated and if they're still doing the same functions that they do before they get injured and after they get injured. So it just depends on how the neurons are working.
Okay, great question. Hi. Okay, Ali and Zella, I see that you have your hand raised. Can you go ahead and unmute yourself and ask us your question?
Yes. How do you donate a brain? Because I think you need brains like to think.
Yes, that's a great question. So some people nobody donates their brains while they're still alive. So some people decide that they want to donate their bodies to help scientists learn and help medical students learn. And so that's something that they would choose to do after they pass away. So they would just make that decision while they're alive. And then after they pass away, they would donate their brain to science. Great question. Max, so we're gonna go ahead and go to some other folks. If you have another question, you can raise your hand again, okay? Max Middleton, your hand is raised. You want to go ahead and ask your question? Hey, Max. What's your question?
How does cancer grow? Or why does it grow faster than other cells?
That's a great question, Max. So cancer cells, they can take up all the food from the environment, so they can eat a lot, and they eat a lot of food, and then they transform that food into new cells. So they double, they become twice their size, and they divide and make 2 cells. And they do that so much faster than any other cell that they can become very large very quickly.
Great question. Okay, Phoenix, I see that your hand is raised. Do you want to unmute yourself and ask your question?
So for the brain cancer, what exactly does chemo do? Does it separate, like I saw on the microscope, it looked like it was separating and decreasing the size of the cancer cells. So what does chemo exactly do to the cell?
That's an excellent question. So different chemo can do different things to the cells, and they can kill cells, they can slow them down. The one that you saw prevents them from growing in size and dividing. What you saw becoming smaller, it's not the cell itself, but it's the factory that makes materials for new cells. So if we stop that factory, the cells don't have the ability to grow and then make more cells. So that chemotherapy specifically prevents the cell growth.
All right. Spooky Science viewer, your hand is raised. Do you want to unmute and ask a question?
Why is cancer
No question. When you do too much medicine for the worm, does it kill it?
So I think that was, let's go ahead and answer that question. Spooky Science, we'll come back to you in just a second if you'll be patient. Thank you. So the question was, if you give too much medicine to a worm, does it kill it?
Mm-hmm. I don't really know. I haven't given my worms too much medicine. I've never killed any of my worms doing that. So I really don't know if that would be the case, but it might be.
I guess it depends on the medicine.
Yeah, it depends.
Okay, Spooky Science viewer, you want to try one more time? I'm sorry, we had to got you cut off there. Do you want to try again? Okay, it looks like, let's switch over to David Gerber then. David, do you want to ask us your question?
Yes. So how do people get brain cancer? Because I know that you can get lung cancer from smoking, but I don't know how like you could get brain cancer if like it doesn't ever like, I don't know.
That's an excellent question. And that's one question we've been trying to answer for a long time. Different people get cancers for different reasons, and we don't know exactly why. But one thing we know is that are mutations that can happen in one cell of the brain, and that cell starts growing faster. So our cells are always talking to each other. But if one cell stops listening to their neighbors and starts growing without listening, look, you cannot grow right here, you cannot grow right now. If they stop respecting their environment, they will grow faster. And there are genes that when they change in those specific cells, they can make that happen. They can make cells grow faster. So there isn't one reason why people get brain cancer. There could be multiple reasons, but they are usually connected to one mutation.
It looks like Phoenix has another question. Phoenix, you want to unmute and ask?
Yes. So how does does a cancer cell specifically bond to like a specific protein, or could it be different cells?
Could you repeat your question?
So if you have like a cell, and how does so they asked like, how does it get in? I'm asking like, is there a specific cell in your body that the brain cancer would bond to duplicate itself?
It doesn't need to bind to anything. They have the ability themselves to make more of themselves. That's why they are so dangerous. So our bodies have specific cells that can make more of themselves. For example, in your skin is shedding every day, and you need to make more skin cells every few days to replenish. But most of our cells don't need to divide because they can live long. But then some cells lose control of that, and then they start dividing for no specific reason, and that's when cancer forms. So those cells have themselves the ability to divide when they should not. It's a mistake that they're dividing, but then that's how cancer forms.
Needy, do you want to unmute yourself and ask your question? I see another raised hand up here.
Is there any organism that can fight off cancer?
That can fight cancer? So there are some organisms that don't seem to get cancer very commonly. There is one interesting rat, the naked mole rat, that seems to live very long and seems to be very resistant to cancer. So scientists are now trying to understand why these animals can have a long life and a somewhat healthy life. But it's not clear why. And I'm hoping that from studying those animals, we'll be able to learn ways of improving our own health and preventing human cancer and cancer in all other animals.
Have you ever tried extracting antibodies from the rat and testing it on a cancer cell?
So we have not, but there are people looking into things that you could learn from the rat. I don't know if it would be possible, but that's a good idea. Maybe someone should look into learning more and more things from those animals and importing into our own healthcare. That's an excellent idea. You should go into science.
We do have one final question, Dr. Dennison, from Alcy. Alcy, if you wanted to go ahead and unmute and turn your camera on and ask our expert your question. Give her 1 second.
So my daughter wanted to ask about the theme of the meeting, brain and bones and fluorescent themes. So yeah, she was curious about the theme.
The theme of the whole event.
I know she's shy. Yeah.
Yeah, so we just tried to pick a Halloween theme. So there's so many cool things in science. It was really hard for us to narrow down what we wanted to show you today. So we picked things that we thought might sort of fit a Halloween theme. So glowing in the dark, and we got these glow sticks, and wanted to talk about brains, and we talked about spine health because we could show you someone doing yoga in a skeleton outfit. So it was just a Halloween theme, but we just wanted to show you lots of cool different science things happening here at UT Southwestern. So thanks for your question, Elsie, and thanks to everyone for your questions. We have a ton of great questions in the chat as well. I want you to know that we have more time for Q&A at the end. So we have a little bit more going on here, but then we have 10 minutes for Q&A at the end, and we'll try and find a way to answer the questions that we aren't able to get to live today. So now we're going to head to Dr. Stu Ravnick in the lab, who will show us how to do some simple, spooky science experiments at home. So Dr. Ravnick, please take it away.
Thank you so much, Dr. Dennison, and happy Halloween again, everybody. I wanted to highlight our shirts that we're all wearing. I am only spooky periodically. If you can't be a fun scientist and also be a little nerdy, you know, you gotta work your way through it. So we're gonna do a little bit of a demo of some kind of science that you can do at home. Now you can see I'm actually here in the lab. This is live. This is the lab of Dr. Jeffrey Woodruff, and he is allowing us to use his space. Now, as in any lab, I want to make sure that I'm clean. So I'm going to put on my Halloween lab coat and make sure that I protect myself. And if I was actually doing experiments in the lab, I'd also put on some wonderful purple gloves, but I'm not going to put them on for this demonstration because I want you to understand that you can actually do this experiment at home and nothing is going to hurt you. So first of all, at home, you might use a bowl, but in the lab, we're going to use a beaker. And we're going to make some slime. Now, we have some glue that we bought from the store. This is just a bottle of clear Elmer's glue. And then we bought some the glow-in-the-dark Elmer's glue. Now, parents, if you want to do this experiment, you can do this with straight glue, but I'm going to actually mix these half and half just because it helps save some of the materials. So I'm going to use 150 milliliters, which is actually 5 ounces. You can use a regular 5-ounce glue bottle. And I'm going to pour the glue, the clear glue, in so that it's approximately half of how much we need. So that's about 75 milliliters. I'm now going to add some of the glow-in-the-dark Elmer's glue, and it's kind of goopy. So I got to really squeeze it out. I hope you can see me squeezing it out into my bowl here. Now, any parents who haven't done this, if you are concerned that this is going to hurt your bowl from your kitchen, I promise you it won't. And you'll see why in just a second. So what we're going to do for this reaction is use some other simple household items. And we actually have this recipe, this protocol set for you on the UT Southwestern website. So now, in the laboratory, we're going to measure things using the metric system, but I know in your house you may not have some of those types of things. So this little baggie has baking powder, baking soda, just regular good old baking soda. And so I'm going to use 5 grams, which is 1.5 teaspoons. So here's 1 teaspoon, and I'm just going to pop it in our bowl here. And then I'm going to use 1/2 teaspoon, pop it in here. Now, glue is a chemical called polyvinyl alcohol, and it's actually a polymer. A polymer is a kind of molecule that has like kind of long chains. So I've used this stirring stick. We usually norm this to bring up liquids, but I'm going to use this to stir. And I'm stirring the glue and the baking soda together. And what the baking soda does for this is it makes it a little thick. It provides the glue kind of a little bit of extra structure. And so it's kind of goopy, it's kind of hard to stir. Now we want to actually make this into slime. And what we're going to produce is what's called a non-Newtonian fluid. And the glue, the polyvinyl alcohol atoms with the baking soda will actually change its molecular structure if you add some boron, some boric acid, the boron ions. And to do that, we're actually using some contact lens solution. If you don't have contact lens solution, you can also use what's called eye wash or anything, as long as it has these boron molecules in it. I'm going to use 8 milliliters, which is a tablespoon. And I'm going to just squirt that in there. And here we go. Add it to our liquid. And this is going to magically, not magically, chemically, it's going to come into the, oh, you can see how it's already getting kind of snot-like and slimy. And oh my gosh, this is wonderful. And I'm going to take this off of my stir stick, get my hand in there and get some of this. Oh my gosh, it's so great. Oh gosh, look at that great. Oh my, oh, I love slime. Slime is so much fun. And you know what's really cool about this slime is that when you mix it, non-Newtonian fluids are so cool because they change when you physically deform them. And so this slime has the property, because of the polyvinyl alcohol, to change its properties so that the more you play with it, the stiffer it gets. But if you leave it alone, it just starts to stretch and grow on its own, which is, I know, it's so much fun. And, you know, kids, make sure you ask your parents to do this. And parents, you can do this with your children without wearing gloves like I am at home. Please do have some supervision. None of this is toxic. And obviously we're not going to eat it because it's not like our delicious brains that Dr. Goldberg has for us. But you can do that. And the nice thing is that the chemicals will actually come away from your bowl. So it all will look really clean. And the nice and fun thing is I made this glow in the dark. Now, I'm not in a dark room, but using this type of glue is really super simple. And you can actually charge it up just using regular light. Now, I brought along a flashlight, which is a black light flashlight. So if I flash this on that, it will actually charge it up. So you can see kind of how it's somewhat glowing a little bit already. And that's the difference. Sometimes you hear of things that are fluorescent, like we heard about from our microscopes, or phosphorescent, or luminescent. These are all kind of the same properties, right? So the glow sticks that we use, those are chemiluminescence, they're making light from chemicals. Whereas this property of phosphorescence is long lasting. So if I charge it with light and I take it to the dark, you'll be able to see that it will glow. You can make that at home. It's really fun. Now, you play with some slime and it gets really kind of cool. You can make long ropes and do all kinds of things with it and it glows in the dark. But how do you make it last? How do you keep it for yourself? Well, here's some slime I made yesterday. I just put it in a baggie and put it in the refrigerator. And you can keep this in the refrigerator and it will last for days. And so you can actually make the slime and enjoy the chemistry, the science of that, what's going on. And then you can have all the fun of being able to play with it. Now, you might ask yourself, okay, this is really cool and it's fun. How do I do other science experiments at home? There are lots of science experiments that you can do at home. And I encourage you, kids and parents, to go ahead and Google, you know, fun science experiments at home. In fact, you can actually squish strawberries with some normal household chemicals, and you can extract the DNA from the strawberries at your house. So you could also be a scientist at home, as long as you're clean and make some fun slime. Now, I had gotten a question about, well, science isn't just for kids, right? It's also for everybody. You know, we'd like to have the adults have an opportunity to have some science. So adults, here's your thing for your Halloween party tonight. Just 1 second. All right, so what I have is another beaker and I have some ice cubes. But these are special ice cubes that I made using tonic water. And tonic water has a fun natural chemical in it called quinine. And quinine has a really fun property that when you shine it under a black light, it will glow as well because the light will activate the quinine molecules and it will glow a little bit. So let's see if we can actually show that a little bit here. So I'm gonna go get some water. I'll be right back. Don't go away. All right, so now you can see that I have my beaker or your glass with some tonic water and just regular water and tonic water ice cubes. And let's see if we can get this to show that it'll glow a little bit. Well, I'm really sorry. It's in the light here in the laboratory, but so you can't really see it glowing too much. But that will be some kind of fun thing for your Halloween party tonight. And I want to make sure that everybody has an opportunity to have a lot of fun during their Halloween and doing their science at home. And I also know that we have lots and lots and lots of questions today. So even though I'm way ahead of my time, and you know, I want to play with my slime, and I know you do too. But why don't we finish our demonstration here and go ahead and move to some Q&A a little early. Would that be all right, Corey and Jenny and team?
That would be great, Stu. Thank you so much. That was a fabulous presentation. Before we get started, we do have a couple more trivia questions that we were going to ask you guys just to learn a little bit more about Halloween. So what we want to know is, what vegetable was originally used as the first jack-o'-lantern? Apple, turnip, onions, or gourds? It's kind of fun because it's kind of unique. We'll give everyone just a couple minutes to answer. Okay, so the majority of you said gourds, but it actually was turnips. Turnips were actually the first thing that we used as a jack-o'-lantern. And moving on to our next Halloween trivia question. So, trick or treat, smell my feet. When did the American tradition of trick-or-treating begin? 1910s, 1920s, 1940s, or 1970s? And again, we'll just give you guys a couple of minutes to get everything answered. All right. So the majority of you said 1940s. It actually started in the US in 1920s, but it started in Europe in actually the 16th century. So trick-or-treating has been going on for a very, very long time. All right, moving on. Okay, our last question before we go to Q&A is, what is your favorite Halloween movie? Is it Ghostbusters, Beetlejuice, Hocus Pocus, or Casper? And personally, you could be watching all of these online right now on Freeform, has them every single day for the last 3 weeks. All right. All right, let's see. So majority of you said Ghostbusters. That is one of my favorites as well, but I have to say Beetlejuice is still my favorite. And one final question. In the Great Pumpkin, Charlie Brown, who does Linus mistake for the Great Pumpkin? Is it Snoopy, Charlie Brown, Lucy, or Franklin? Let's see how many of you have watched that amazing classic. All right, so let's see. Majority of you said Snoopy, which is correct. Nice job, everybody. We are now going to move to questions. So if you guys want to go ahead and utilize that raise your hand function again, we will call on you to unmute yourself and ask your question. So our first question comes from Jennifer Pope. Jennifer, do you want to go ahead and unmute and turn your camera on?
Yes. Are people close to finding a cure for cancer?
That is a fantastic question. We will go ahead and pass to Mara to answer that one for us. For just a second. Dr. Mara-Lise Kanachi, can you hear us?
I wasn't able to unmute myself.
No problem.
That is an excellent question and a very difficult one to answer. So we are working very, very hard. Many of us, we're all working together and very hard to try and find not only the cure, but ways of preventing cancer. Each cancer is a different disease. We call them all cancers, but they are cancers that appear in different organs, are very different from each other. And it's our belief that there won't be one cure for cancer in general, but we're going to have to find a cure for each specific cancer. And sometimes the same type of cancer between two people are also different from each other. So we're developing more and more tools and medicines that will help treat patients. But the cure will come to specific cancers at specific times. And it's a wonderful question. And we need more scientists working to try and help us find cures to cancers and to many other diseases.
Thank you.
Okay, so moving on to our next question comes from Nidhi. Nidhi, you have a question if you want to go ahead and unmute yourself.
How does black light charge the glow-in-the-dark slime?
That's a great question. Dr. Stu Ravnik.
So I've moved out of the lab to a little office here, so the science can going. That is a great question. So what happens with the glow-in-the-dark slime? That glue has extra chemicals put into it, and so what happens is, like I said, fluorescent versus phosphorescent. So fluorescent means that it only shines very quickly and very brightly for a short time. You have to have the special black light. But phosphorescent, like the slime with the glow-in-the-dark glue, actually you can charge it with light. And what happens is the energy in the light, I'm pointing to the light in the ceiling here. The energy in the light with the photons will go into those chemicals and will cause the molecules to jump around a little bit. That's not exactly, but it's an analogy. So when those molecules jump, they jump from one energy area to another energy area, and that allows the light, the phosphorescence, to show. Great question. I love slime.
Thank you, Dr. Ravnik. And we did just get a comment that everybody loves your hair. So moving on to our next question comes from Max Middleton. Max, do you want to go ahead and unmute yourself?
How does the slime glow when it shines the light?
So great
Or why
question.
does
Oh yeah, so this slime glows because I used the special glow-in-the-dark glue, right? If you just make normal slime with regular glue, it's not going to glow in the dark. So this glow-in-the-dark glue has the special chemicals in it, so that when you charge the light, it makes those energy jumps, and those energy jumps allow it to have that. And that's exactly how it works with what we saw in the microscope. Instead of having regular light, we use special light in the microscope to make those worms glow or the cancer cells glow. It's a great question. Thank you so much.
Thank you.
Thank you, Dr. Ravnik. Thank you, Max. Our next question comes from David. David, do you want to go ahead and unmute yourself and ask your question?
Never mind.
Okay. Our next question comes from Vega. Vega, do you want to go ahead and unmute yourself and ask your question?
What is the activator in the slime and how does it help the slime stick together?
I love all the questions about slime. Chemistry is so much fun. So the activator, so if you buy the chemicals, you can actually buy glue and you can buy what they sell like on Amazon or in a craft store, what's called activator. And that activator already has in it the mixed in either starches or baking soda with some kind of solution that has these boron atoms in it. That's the activator. What I did in the laboratory is I made them from home stuff. So I added the baking soda separately and I added the contact lens solution separately. Now you can actually use other materials to make your own activator at home. They're just not as nice on your hands. So like some of the laundry starches and things like that. And so I put some of those things in the information on the online documents. And that's a great question. Thank you so much. I also saw a question of someone asking, can you use hand sanitizer to make slime? No, you cannot. Hand sanitizer has alcohol. That will actually break these polymer bonds. So You can use your hand sanitizer to clean hands and stay safe during COVID, but not for slime. Great questions.
Great, thank you. Our next question comes from Amanda Hendrickson. Amanda, if you want to go ahead and unmute yourself and ask your question.
Do animals and humans' brains work the same?
That is a great question. Dr. Goldberg, can you answer that question for us? Dr. Goldberg, just a second. Dr. Goldberg, you are muted.
Okay, a wonderful question. Thanks so much. Absolutely, we work, animal brains and human brains are very similar in many, many ways. We share much of the same DNA and many of the same genes as many animals. So I have a cat here next to me, and that cat and I share many of the same ideas about how the brain works and how things process. But of course, there are many things that our brains do that my cat's brain can't do. And of course, our brains have evolved to do additional things. So we have additional areas in language and in coordination and in cognitive thinking and how we remember things that a cat might not have or a mouse might not have. So they're very similar in some ways. And if you're thinking about a disease like cancer or a brain disease, maybe they're similar enough to help us understand some of the diseases that we study, but maybe not as similar as they need to be. So when we want to study a real serious problem in neuroscience or a real serious problem in disease, we have to study both people and animals to be sure we're getting the entire picture. And it's a great way to ask the way you did. Thank you.
Thank you, Dr. Goldberg. Our next question comes from Violetta and Grandma Pat. You guys have a question.
How do you make the strawberry slime?
Dr. Ravnik, can you talk a little bit about the strawberry you were talking about?
Thank you so much. So what I was talking about with the strawberries is you can actually get some DNA from those strawberries at home by squishing them up and then adding some chemicals. And so we will try to add the recipe for that. But if you just Google, how to extract or make DNA from the strawberries, you can find that, how to do that at home. You might have to buy a couple of special things, but it's really, really cool. And the reason we use strawberries is because those strawberry fruits have many times the normal amount of DNA in their strawberry cells than normal humans do. So you can really get some really good gooey DNA from that.
Pokemon last around today.
Thank you, Dr. Ravnik. Just a reminder to leave yourself muted until you're called on. Oh, thank you for the question. We are going to move on now to Sophie Kittner, our good friend Sophie. If you want to go ahead and unmute yourself and ask your question.
Oh, you want me to ask it? Okay, Sophie wants me to ask it. She'd like to know,
Kittner, whichever works.
Yes, she'd like to know if Dr. Ravnik or anybody has good recipes for edible slime.
That is funny how you asked this, because someone asked about strawberry slime. Yes, there are recipes for edible slime. I don't have them with me, and I don't remember them, but there are ways that you can do it. You can't use the same chemicals I use today because you don't want to eat that. Now, you saw Dr. Goldberg earlier have his brain that he ate. This is kind of a Jello brain that's kind of slimy. And Dr. Goldberg, that is a good brain.
Is that what it's actually made out of?
No, this one's actually made out of Jello, and so, but it's so good. Nom.
Thank you, Dr. Ravnik.
And thank you, Sophie, because I remember you asked the very first question at our very first Science Cafe. So I'm so happy to see you here, and I love your costume.
Show them your slime.
Oh, and slime. Awesome. We're moving on to our next question from Vega. Vega, do you have a question? Go ahead and unmute yourself. Just a second.
I have a question about how does your spine help you walk?
That is a great question. And we are actually going to pass to our Frisco spine team to actually answer that question. So we are all together in the Frisco Spine Lab.
That's a great question. Our spine helps support the weight of our whole body. So as we stand upright, little bones that make up our neck, middle part called thoracic and lumbar, all work together to bear our weight so that our legs can move us forward.
Thank you. That was a great, and if we have time for probably one more question, we have seen a couple of questions in the chat box, but if we could also raise our hand. Dr. Goldberg, could you talk about how training your brain not to react to specific things, other parts of the brain besides the part that controls fear?
Okay, so if we are
Dr.
afraid
Goldberg,
of things,
you are muted.
Not muted. Can you hear me now?
Give you just a second, and we'll get you unmuted.
He's
Right.
not muted, Corey. We can hear him.
I'm not muted. I am talking. I just have little distractions here that I have to deal with. So, okay, so imagine that if you're worried about it, if you're afraid of things, certain times, if you're afraid of cats, maybe the way to get to be not afraid of cats is to see a cat, or maybe to get closer to a cat. And more the times you expose yourself to something that you're frightened of, your brain learns eventually it's not a threat, and maybe cats are okay, or maybe it's jellyfish you're afraid of, or worms. And oftentimes, in the normal course of affairs, our bodies and our brains learn that things aren't threatening because we see them and we learn that there's not going to be a threat. Sometimes it just doesn't work that way. Sometimes there's things that we're just afraid of and we're not going to not be afraid of them. And no matter how much we try to expose ourselves to those things, it's not going to change because our brain's just not capable of making that change. And it's different for each person, the kinds of things that we might be afraid of. And in fact, when we watch horror movies, they take advantage of those things. Certain things just everybody's afraid of, and that's what makes for a scary movie. So I think the question about how our brains learn which things we should be afraid of and which things not to be afraid of has to do with our experience and with the connections we make. If you've been stung by a bee, it'd be a good idea to know that bees are something to be afraid of, but maybe not too afraid. So I hope that answers the question. It's a really interesting area of science, how we make those connections and how those connections of things we learn tie into our emotions, what we feel about what we know. And that's a big part of how, a big question that scientists study just about every day.
Well, thank you, Dr. Goldberg, and thank you to all of our experts who joined us today. I'm actually going to pass it to my colleague, Jenny King, to close us out.
Hey everyone, thank you. Those were the most excellent questions. You kids are so smart, and all of us adults, I know I learned a lot today. I might try slime again at my house with my own kids. And if anyone wants to try that recipe, they need to go to www.utsouthwestern.edu/spookyscience. If everyone who worked on today's program, all my wonderful colleagues, thank you. Wave while we're closing out. Want to thank the Dallas Morning News for this opportunity and for including us year after year. Thank you everyone for being on today. We appreciate your interest in learning about science. We know it's fun, it's cool, and it is super important. We will be sending you an email survey. I know everyone loves those. Fill them out. Let us know what you want to learn about at the next Science in the City. Be safe out there, guys. Wear your masks, even when it's not Halloween. Stay home when you can. Stay 6 feet apart. Wash your hands really, really good. And we hope you have a great and ghoulish rest of your Halloween. And we are adjourned.
Always thank you, Dr. Ravnik.
Oh.
Show them what you're making right now.
What you
making slime.
Oh.
We have glue, contact lens solution, and what else, Sophie?
A teaspoon.
A teaspoon of what?
Contact lens.
Oh, it's all you need is the glue and the contact lens solution, and that's it?
Baking soda.
Baking soda.
Baking soda. Yeah.
And Jeff, if you go to the link that we put in the chat, which we're also going to email to you, There is the recipe for that, as well as the recipe for the Jello brains and some links to some other Halloween science.
There's a recipe for the Jello.
Should we make the
brain?
Jello brains?
Sophie, there
We're gonna make the Jello brains.
I you are.
Everyone, you guys,
Wait, how much baking soda do you need?
Use 1.5 teaspoons.
1.5 teaspoons. You got that?
Here.
Okay.
1.5.
I'll get the baking soda, but don't get it on the computer.
And how much contact lens solution?
Use 1 tablespoon or 4.5 milliliters.
1 tablespoon.
Now she just put the whole jar of glue in.
Yep.
Okay.
That's fine.
Now just make sure to not over the computer, so.
what color are you doing, Sophie?
Making slime?
So what color? Is it clear slime?
Yeah.
We've made so much slime over the last 8 months.
I might have some slime residue in my house as well.
That's good.
Wait. Oh yeah, 1.5.
All right, so now you've done that.
Baking soda, Dad.
All right, I'm gonna get the baking
Okay.
soda.
And now we need to have some Halloween jokes. So we know that zombies like to eat brains, right?
Yeah.
What do vegetarian zombies eat?
What?
Grains.
I'm a vegetarian. I like my grains.
What is a zombie's favorite way to travel? Trains.
Most do.
Okay.
Half a teaspoon of baking soda.
Half a teaspoon of baking soda.
1.5 teaspoons.
Yeah, half
Okay.
a teaspoon.
We're not gonna do it.
1.5 teaspoons. Yep.
Oh, 1.5.
Oh, 1.5.
So if we're gonna do 1.5. Let's do
Question while we're making slime together.
Allison Harrison has her hand raised. Allison, you want to ask your question?
Daddy, there's a real 1.5.
No, here's one.
Okay, so it's actually Addie, and Addie has a question.
Let me do another.
Say it out loud, Bug.
How do you make the Jello look like a real brain?
Okay, Eddie, what a great question. And you're absolutely right. If you just make Jello, it's going to be in the same shape as whatever you cooked it in, right? And so the trick here is you have to have a hat, a little mold that is exactly the shape of the brain. I'll be right back to show it to you. Stay there. Don't go away. Don't go away. Where's my mold? All right.
Dr. Goldberg's cat is disappearing in and out. Must be a Schrodinger's
We're
cat.
getting extra help here. And you can buy this on Amazon or anywhere else. Look for Jello brain mold. And all you do is you make your Jello. You added some evaporated milk, non-fat evaporated milk, and some food coloring. And pour it in there, put it in the refrigerator overnight. And you will take it out very carefully. And out will pop your delicious brain, whatever color you want it to be. If you want it to be kind of brain-like, you can make it with peach Jello. But you can pick any color you like. And if you add a little evaporated milk, it makes it look kind of like a real brain. Okay, you got it? Next Halloween, I want to see some brains.
I'm making some. Thank you.
Amber had a great Halloween joke in the chat that everyone should go check out.
Do a Halloween joke.
Birds say on Halloween, trick or tweet. That's really good, Amber. I like that.
That should be on Twitter. We should tweet that.
Twitter.
Like that spectacular day.
Why was the skeleton so afraid of Halloween?
Why?
He didn't have any guts.
I made slime.
Show them your slime.
My slime is so clear.
That's so cool, Sophie. Thank you.
That is very cool slime, Sophie. Well, I am going to go ahead and close us out. But again, thank you all for joining us. We hope you have a wonderful and safe and socially distant Halloween today. And as Jenny said, please keep your masks on and everyone stay safe. And everyone have a great day. Happy Halloween.
Bye.
Happy Halloween.
Bye, everybody.
Bye.